In this video, we will discuss what is recombinant DNA technology, what are the various vectors which are involved in DNA technology, types of vectors, and properties of vectors. Now, first we'll discuss what is recombinant DNA technology. This is also called as genetic engineering. So we can define it. This is stream of biotechnology which deals with manipulation of genetic material by man in vitro. Now, man is always interested in high quality of uh, crop materials, animals. So he keeps on manipulating the genetic material of plants and animals. And this manipulation or changing of genetic material is called as genetic engineering. For example, if man wants to increase the yield of certain plants, so man will isolate a DNA or gene of high yield and incorporate into our domesticated varieties to improve the yield. Similarly, if a man wants to improve the breed of the animals or certain other properties like egg laying capacity, milk yielding capacity, so a man will isolate a gene from animals or chicks which have high egg laying capacity and animals which have high milk yielding capacity, and that will be incorporated into our domesticated animals. So in this way he can manipulate the genetic material in the lab, and this technology is also called as biotechnology. And this manipulation, that is insertion of DNA into the host and combining two kinds of DNA, and then if that DNA shows its expression, then the man is successful, and then whole process is called as genetic engineering. So DNA, first it has to be isolated. So that isolated DNA is called as gene of interest. I mean, we are interested in that gene. Now that gene will be cut. Supposing this is DNA, so this part will be cut, right? So this is called as gene of interest. Now this gene of interest cannot be cut every time. So this can be multiplied. Gene of interest has to be multiplied. Its multiple copies are produced, and this multiple copy production is called as cloning. So these copies, they will be completely identical. Many copies, million copies, thousand copies can be produced, and this process is done with the help of cloning process. This cloning we will discuss in another video in detail. So recombinant DNA is equal to vector plus insert. Now what is vector? Basically, vector is the vehicle, cloning vehicle we can say, which is used for multiplication of the DNA, that is our gene of interest, or insert DNA. This DNA is also called as insert DNA. Now this can be put into the vector, it can be multiplied, and multiple copies can be produced, and then this is called as recombinant DNA. Now we can see in this figure, supposing we are using the vector plasmid, right? Plasmids, basically they are present in bacteria. Now this is the bacterial genome, but this is the extra chromosomal DNA called as plasmid. Now this plasmid is basically double stranded. It has two strands of DNA, and this has certain sites for restriction sites. Restriction sites mean it can be acted by restriction endonucleases. Now these are specific enzymes which can cut the DNA at specific points. For restriction endonucleases, I have shared the link in the description box. You can watch that video. What are restriction endonucleases? A plasmid may have multiple restriction sites. This may have one restriction site. Now this is the point which is showing ORI point, mean origin of replication, because this plasmid DNA can independently replicate inside the bacteria. Supposing this is bacteria, this is plasmid, this is the bacterial DNA, but this plasmid will replicate independently. This is independent of replication of the bacterial DNA. So this point is called as ORI point, that is origin of replication. Now this is the DNA which we want to insert. Now this can be again cut with the help of restriction enzymes. These are specific site for cutting the DNA. So this DNA we have cut and we want to insert here into the plasmid. So this will be cut with the help of restriction endonucleases. A cut will be generated and here we can insert our DNA. Now we can see this is a recombined DNA. This is not normal plasmid. Now this has gene of interest over here. So this is called as recombinant DNA. Now we will multiply and make multiple copies of this DNA. Then we can insert this in DNA of interest or gene into the host cell, and if it shows its expression, then we are successful in the genetic engineering experiment. So this is how genetic engineering is done. A DNA is cut from somewhere and that is put into the cloning vehicles for multiplication. Then after multiplication, that is put into the host where we want to insert that DNA, and this is called as recombinant DNA technology, or this is called as genetic engineering. For the whole process of genetic engineering, there are certain tools which are required. So what are the tools of genetic engineering? One, we need to have restriction endonucleases. They are specific enzymes which will cut on specific DNA sequences. Again, a link is shared in the description box. Then second, we need to have vectors for cloning the gene of interest or DNA. We want to make multiple copies because every time we cannot cut the DNA. So first we will cut the DNA and then we'll create its multiple copies. And for this whole process, for creation of multiple copies, for cutting the DNA, we need to have different kinds of enzyme. Supposing we said this is the plasmid and we want to insert here, supposing this is the plasmid and we want to insert here this DNA, right? So one, we said this can be done with the help of restriction enzyme, right? Now this DNA has to be combined with this DNA, right? Then we need to have enzyme DNA ligase. So there are many other kinds of enzyme that we'll discuss in the next video, which enzymes they are required for use in the genetic engineering. So various kinds of enzymes they are required. Then we need to have foreign DNA or gene of interest, mean DNA, which we, supposing this is DNA, we cut the DNA and we take this DNA, right? This foreign DNA or gene of interest, this has various names. This is called as gene insert. This is called as gene of interest. This is called as foreign DNA. We can give different names to it because we are interested in this DNA. So this is called as foreign DNA or gene of interest. Then we need to have probes and linkers. This again we'll discuss in detail. I will not go into the detail of probes and linkers here. So there are various tools like restriction endonucleases, vectors, various enzymes, foreign DNA, probes and linkers. Restriction endonucleases, I have shared the link in the description box. Vectors we'll discuss in the next video. Then we'll continue with all other factors like various enzyme, foreign DNA, probes and linkers one by one, and you can watch my videos. Now we first will see what are vectors. As we discussed, vectors, they are vehicles which are used for cloning of DNA segment in the cell. What is cloning? Making of multiple copies and totally identical copies of one DNA. This is called as cloning. So a DNA molecule acting as cloning vehicle should have following properties. If we are using certain vectors, they should have certain properties or rules to follow. And what are those properties? This we will discuss. Now we are taking here example of plasmid. But there are certain other vehicles also. It's not only the plasmids are used as cloning vehicles. Now this has double stranded DNA. Then it has a point which is called as ORI point, or this point is called as origin of replication. What does it mean? Origin of replication. Mean this can independently replicate. It is not dependent on the bacterial genome. This will independently replicate. Similarly, when we are using other vectors also, they should have this point which is called as ORI point. They should be independently replicating. Then every vector should have a marker gene. A marker gene can be antibiotic resistant gene, this can be nitrogen fixation gene, this can be toxin production gene. What will be the advantage of this antibiotic resistant gene? Supposing we have a culture of plasmids. I'll make here with the help of diagram. I'll explain to you. This is a petri dish, right, in which we are culturing the bacteria. Supposing these are the bacteria which we are cultivating, right. Now, if supposing this is one bacterium, right, this has a plasmid here, and this plasmid has a gene here, which is called as antibiotic resistant gene. Supposing this is resistant to tetracycline, right, and in this petri dish, when we are adding the nutrient medium here, when we are adding the nutrient medium, we are adding the nutrient medium with this tetracycline antibiotic. Now, only those bacteria will survive which have this marker gene, that is antibiotic resistant gene, right. All other bacteria, they will die because this has a gene which can resist the antibiotic called as tetracycline. So we can see here, now these bacteria which has this gene will survive, and all other bacteria, they will die. This will die. They will not survive, right. Only with yellow gene or marker gene will survive, and all other bacteria, they will die. So this should have a marker gene which can be antibiotic resistant gene or any other gene, right. Then it should have multiple cloning site, like it can be cut with the help of restriction enzyme at various point, and this can be cloned in a various ways. This is called as multiple cloning site. Then a strong promoter. Then where the cloning vehicle which we are using, it should be easy to isolate and purify. It should not be complex that we have to follow a very complex procedure for isolation and its purification. So supposing we have this cloning vehicle here, this should be easily isolated and purified, easily handled. It should not have a complex DNA also. It should have a very simple DNA or simple genome. Then it will have various kinds of restriction sites which can be acted on the plasmid. Supposing this is plasmid, so if it has only one kind of site for the restriction enzyme, then it becomes difficult. It may have different restriction sites where we can put our DNA or gene of interest. So these are the properties when we are using certain vectors. So we should keep in point these properties, that vectors should have these properties, that is, it should have ORI point, it should have a marker gene, it should have a multiple cloning site, it should have a strong promoter for the expression of the gene, then it should be easy to isolate and purify, it should have a simple DNA sequence, and it should have multiple or unique restriction sites which can be used for cutting the DNA. Next is what types of vectors we can use for the genetic engineering. One, we can use plasmid as we have discussed. Second is phages. Phages are basically, supposing this is bacteria and this is the virus which is parasitizing the bacteria. This is the viral DNA. So these viruses which parasitize bacteria, right, they are called as phages. Viruses parasitizing bacteria, they are called as phages. So this viral DNA, this can be multiplied, or this can be cloned, or this can in this we can put our gene of interest over here. We can insert the DNA over here, right, and we can multiply with the help of viruses. This is called as phages. This we'll discuss in the next videos. The next is cosmids. Cosmids basically, they are combination of plasmids and phages called as cosmids. Next is phasmids. Again, they are combination of phages and plasmids. Then shuttle vectors, which can be used in two hosts. Then yeast vectors. A yeast is a basically unicellular fungus, and its DNA can be used for construction of yeast artificial chromosome called as YARCs, which can be used for the genetic engineering program. Now next is we can also use transposons. I have shared a video of transposon in detail. What are transposon? Basically, they are jumping genes which can jump from anywhere. Supposing this is DNA. So this DNA may break up and this may get combined somewhere else. So this is called as transposons or jumping genes. I have shared a link what are transposon in the description box. You can see that, and how these transposon can be used as genetic engineering. This will discuss all the vectors. Will discuss one by one: plasmids, phages, cosmids, phasmids, shuttle vectors, yeast vectors, and transposons. So this is all about this video. In the next video, we'll discuss vectors one by one. Like first, we'll discuss plasmids. So thank you for watching my video. If you like my video, please like, share, and subscribe.